Hello and welcome to Med Simplified. In this video, we will discuss about the Krebs cycle. I posted a brief video on the topic of Krebs cycle a few years back, and that was a hugely popular video among you guys. So this will be an update to that video in which we will discuss Krebs cycle in more detail but in a simplified way. At least I'll try to. So let's begin. Krebs cycle is also known as the Hans Krebs cycle or the citric acid cycle. Now, Hans, after the scientist who discovered it, Hans Adolf Krebs in 1937, and received a Nobel Prize for Physiology and Medicine in 1953. Now, this cycle is also known as the citric acid cycle after the molecule that plays the central role in Krebs cycle, the citric acid also known as the citrate. The citric acid cycle is a key metabolic pathway that connects carbohydrate, fat, and protein metabolisms. The TCA cycle is a central pathway into which many metabolites feed. It consists of a number of reactions which generate NADH and FADH2, which are in turn used to generate ATPs. The Krebs cycle consists of a series of chemical reactions that release stored energy of fats, carbohydrates, and proteins through the chemical process of oxidation. Other than being the central pathway for generation of energy, the Krebs cycle is also very important as it provides many raw materials that are used elsewhere in the body, like amino acids and NADH. An interesting fact about the Krebs cycle is that it was one of the earliest established components of cellular metabolism and may have originated abiogenically, meaning it originated even before the life started. This is the concept of abiogenesis, that the life originated from non-living things. Get the lecture handouts from this video and all other videos on Med Simplified. Join our community at patreon.com or click the join button below. Now, before diving into all the chemical names, the enzymes, and the various steps of Krebs cycle, I would like to take a few minutes to talk about the conceptual basis of Krebs cycle that most teachers fail to explain. So, like I told you, Krebs cycle is a central metabolic pathway. Now, what do you mean by a central metabolic pathway? Now see, when we eat food, it is digested, absorbed, and is first processed by initial metabolic pathways. These initial metabolic pathways generate some common products that are used by the central pathway, like the Krebs cycle, to produce energy. Like if we take the example of carbohydrates, carbohydrates are digested into simple sugars like glucose, and the glucose is first processed by initial metabolic pathways like glycolysis. Glycolysis produces pyruvate, and similar products are produced by the initial metabolism of fats and proteins as well. The pyruvate is then fed into the central pathway, that is the Krebs cycle. So the Krebs cycle provides a central pathway that connects carbohydrate, fat, and protein metabolisms. Now the next important concept is how does the Krebs cycle produce energy? For this, let's take the example of metabolism of carbohydrates again. We get simple sugars at the end of digestion of carbohydrates like glucose, right? And after this, glucose undergoes through all the metabolic pathways like glycolysis, Krebs cycle, and electron transport chain. One molecule of glucose produces approximately 36 ATPs. But where does this energy come from? The single-worded answer to this question is through the chemical process of oxidation. Now see, what happens is that through glycolysis and an additional reaction, we break down glucose into small 2-carbon fragments which are known as acetate. This acetate is then fed into the Krebs cycle. If you look at the Krebs cycle, it consists of these 10 intermediate molecules that start and end with citrate. When we add acetate to this cycle, this is a 2-carbon compound. This cycle takes a full turn and releases 2 molecules of carbon dioxide as a waste product, meaning the 2 carbons that we added in the form of acetate are released in the form of carbon dioxide. This carbon dioxide is produced due to the process of oxidation that occurs in the Krebs cycle as a result of various enzymes. Electrons are added to NAD to make NADH and FAD to make FADH. These electrons are then subsequently used in the electron transport chain to make ATP through ionic gradients. Now all of this may seem a bit confusing in the beginning, but my advice is to take things slowly. Watch the other videos regarding glycolysis, electron transport chain and metabolism of fats on our channel to have a complete understanding of this metabolic pathway. The links to all these videos are in the description below. Meanwhile, let's begin with the details of the Krebs cycle and various enzymes involved. So in eukaryotic cells, the citric acid cycle occurs in the matrix of the mitochondria. In the prokaryotic cells such as bacteria, which lack mitochondria, the citric acid cycle is performed in the cytosol. Now there is a very important reason why a Krebs cycle takes place in the center of the mitochondria. This is because of the presence of the electron transport chain in the membrane of the mitochondria. This electron transport chain is a chain of many different enzymes that uses the products formed in the Krebs cycle to produce energy in the form of ATP. Now to learn more effectively about the Krebs cycle, it is very important that you have a little bit of background knowledge about the preceding steps that happen before Krebs cycle. So the Krebs cycle begins with this molecule, acetyl-CoA. But where does this come from and what is its role in the Krebs cycle? This is very important to understand. Now one of the important biochemical pathways that runs before the Krebs cycle is glycolysis. In this pathway, we break down glucose, which is a 6-carbon compound, into 2 molecules of pyruvate, which is a 3-carbon compound. What happens next is that this pyruvate is acted upon by a set of enzymes collectively known as the pyruvate dehydrogenase complex. In this reaction, coenzyme A is also involved, which is a coenzyme derived from vitamin B5. In this reaction, 2 carbons are transferred from the pyruvate to coenzyme A to form the acetyl-CoA. One additional carbon present in the pyruvate is lost in the form of carbon dioxide. In this reaction, 1 molecule of NADH is also produced. So this is how acetyl-CoA is produced from pyruvate. This acetyl-CoA is a 2-carbon compound and is the starting point of Krebs cycle. In this molecule, the coenzyme A, the part which is derived from vitamin B5, acts just like a carrier which transfers the 2 carbons attached to it to the Krebs cycle. So the first step of the Krebs cycle is the combination of acetyl-CoA with oxaloacetate. Like I told you, acetyl-CoA is a 2-carbon compound, and this 2-carbon compound combines with oxaloacetate, which has 4 carbons. This combination leads to formation of citrate, which is a 6-carbon compound, and this reaction is made possible by the enzyme citrate synthase. The name of this enzyme is self-explanatory for what it does. The citrate then undergoes a series of enzyme-catalyzed reactions in which it loses the 2 carbons of acetyl-CoA in the form of 2 molecules of carbon dioxide. The conversions, which involve up to 10 chemical reactions, are all brought about by enzymes which use the energy generated through oxidation to attach electrons to NAD and FAD. Now let's look at what happens next with citrate. In the next 2 reactions, the citrate is converted into its isomer, which is known as isocitrate. Both these reactions, which I have shown with just 1 arrow, are catalyzed by the enzyme aconitase. Now this is an iron-containing enzyme which reorganizes the atoms of citrate to form an isomer isocitrate. Now you may ask, what was the need to convert citrate into isocitrate? Why can't the citrate just move forward with the pathway? Well, the answer to that is that the arrangement of atoms in the isocitrate is better suited for the next few oxidation reduction reactions that are about to come in the Krebs cycle. In the isomerization of citrate to isocitrate, an intermediate is also generated which is known as cis-aconitate, but no need to go into the details of that. Moving on to the next step in the Krebs cycle. In the next step, the isocitrate is converted into alpha-ketoglutarate by the enzyme isocitrate dehydrogenase. Now we come to the first of the 4 oxidation reduction reactions in the citric acid cycle. These oxidation reduction reactions are the one which produce energy in Krebs cycle. And all of these 4 reactions use enzymes in them which are known as dehydrogenases. Now, what do these dehydrogenases do and how do they produce energy? That is very important for you to understand. See, if you look at the term itself, dehydrogenase, this literally means removal of hydrogen. So dehydrogenases are basically enzymes that remove hydrogen from a molecule. Now, as you remember, that removal of hydrogen is also known as oxidation. And the hydrogen that is removed from one molecule must be passed on to some other molecule. The other molecule that receives the hydrogen gets reduced in return. So this is the basic mechanism of action of dehydrogenases. They remove hydrogen group from one molecule, oxidizing it, and in turn attaching that hydrogen group to some other molecule, leading to its reduction. This is why the reactions carried out by dehydrogenases are known as oxidation-reduction reactions. So coming back to our Krebs cycle, we reached a point in Krebs cycle where isocitrate will now be acted upon by the enzyme isocitrate dehydrogenase. So let's look at what happens in this reaction. This is actually a two-step reaction in which the enzyme isocitrate dehydrogenase first removes hydrogen from isocitrate, thus oxidizing it. The hydrogen that is removed is attached to NAD to form the NADH, thus completing the process of oxidation and reduction. But there is a small additional step that happens next, which is catalyzed by the same enzyme isocitrate dehydrogenase. In the next step of the reaction, a carbon atom is also removed from isocitrate in the form of a molecule of carbon dioxide. This leads to the formation of a 5-carbon compound known as alpha ketoglutarate. So if you look at the complete chemical process that just happened here, this is known as the oxidative decarboxylation. Oxidative because oxidation of isocitrate takes place since a hydrogen is removed from isocitrate, and decarboxylation as a carbon group is also removed from isocitrate. The conversion of isocitrate into alpha ketoglutarate is followed by a second oxidative decarboxylation reaction, the formation of Succinyl-CoA from alpha ketoglutarate. This reaction uses the enzyme alpha ketoglutarate dehydrogenase that catalyzes a similar oxidative decarboxylation of alpha ketoglutarate. In this reaction also, a hydrogen gets attached to NAD to form the NADH. Since it is also a type of decarboxylation, a carbon is removed from alpha ketoglutarate to be lost in the form of carbon dioxide. Also, coenzyme A is used in this reaction, therefore the product will be a combination that is known as Succinyl-CoA. In the next step of the Krebs cycle, the Succinyl-CoA is converted to succinate. This reaction is catalyzed by the enzyme known as Succinyl-CoA synthetase. Now try to understand what happens here in this reaction, as this is a very important concept and often missed by many students. The number of carbons remain the same in this reaction. Both Succinyl-CoA and succinate have 4 carbons each. So what changes? Succinyl-CoA is a high energy compound, and the energy stored in this compound is used to attach an inorganic phosphate molecule to GDP. This GDP gets converted to GTP, which is like a close cousin of ATP, and is later used in the formation of an ATP. The coenzyme A attached to the Succinyl-CoA also gets released in this reaction. So what happens here essentially is that the Succinyl-CoA loses its high free energy and its coenzyme A to be converted into succinate and a molecule of GTP. This is an example of substrate level phosphorylation, a process of producing energy that we discussed in detail in our glycolysis video. So now we have 4-carbon compound known as succinate. In fact, the number of carbons do not change from here onwards. All the compounds which come next in the cycle only have 4 carbons. The last compound generated in the cycle is oxaloacetate, which also has 4 carbons. And that oxaloacetate combines with another molecule of acetyl-CoA to form citrate, a starting point of the Krebs cycle. Coming back to where we left the cycle. So we have succinate, which is a 4-carbon compound, and it is acted upon by the enzyme succinate dehydrogenase. As you can tell from its name, this enzyme is a dehydrogenase. So it removes hydrogen from succinate, and the hydrogen that is removed is accepted by flavine adenine dinucleotide or FAD. FAD is in turn converted to FADH, and the succinate gets converted to fumarate. FAD is the hydrogen acceptor in this reaction and not NAD, because attaching hydrogen to NAD requires more energy than this reaction can provide. So FAD is used here as it requires less energy change to get reduced to FADH. Free energy change for 4 carbon compounds is less as compared to 6 or 5 carbon compounds that were present at the first half of the cycle. There is, however, an exception to this at the end of the cycle that we will discuss in a bit. Now, the enzyme involved in the conversion of succinate to fumarate is succinate dehydrogenase, which, like aconitase, is an iron-sulfur protein. This enzyme is also very important as succinate dehydrogenase is directly associated with the electron transport chain. The next step in the Krebs cycle is a hydration reaction, which means we add a molecule of water to fumarate and convert it into malate. This reaction is catalyzed by the enzyme fumarase. Nothing special in this reaction, so let's move on to the next one. The next type of reaction that happens here is the oxidation, in which we finally oxidize malate to oxaloacetate. And the enzyme involved here is again a dehydrogenase, which is known as the malate dehydrogenase. Again, the NAD molecule gets reduced to NADH, which will be used in the electron transport chain. In this reaction, NAD gets reduced to NADH. Now you may ask why NAD gets reduced here and not FAD, because like I told you earlier that the free energy change for compounds in the end of the cycle is less as compared to that was present at the first half of the cycle. Well, this was the exception that I was talking about. Even though this reaction is present at the end of this cycle, still a molecule of NAD is getting reduced here. Well, this is due to the fact that oxaloacetate that is being produced here is rapidly being utilized by combining it with acetyl-CoA to form citrate. And the second product being produced here, the NADH, is also being rapidly used in the electron transport chain. So the standard free energy for this reaction, unlike that for the other steps in the citric acid cycle, is significantly positive. I know that some students, especially in lower grades, may find some of the concepts I explain here a little advanced, but trust me, you will have to learn about the cycle in depth at one point or another, so better now. The oxaloacetate again combines with a molecule of acetyl-CoA and leads to the formation of citrate with the help of the enzyme citrate synthase. A water molecule is also added in this reaction. Now this completes one full turn of the cycle and the same sequence repeats again. Okay, so if we look at the cycle, now you can see that we got what we got from this cycle. We added 2 carbons to this cycle in the form of a molecule of acetyl-CoA. These 2 carbons were released in the form of 2 molecules of carbon dioxide here and here. In this process of oxidation and reduction, we convert 3 molecules of NAD to NADH, 1 molecule of FAD to FADH, and we converted 1 molecule of GDP to GTP. Now there are few other important concepts regarding the Krebs cycle that you must understand to have a complete knowledge about this biochemical cycle. The first is this summary reaction of this cycle. And the second is the energetics of this cycle, meaning how many ATPs are produced for every 1 molecule of glucose that enters this cycle. So first, coming to the summary reaction of glycolysis. Now the net reaction of citric acid cycle is 1 molecule of acetyl-CoA plus 3 molecules of NAD plus 1 molecule of FAD plus 1 molecule of GTP plus 1 molecule of inorganic phosphate plus 2 molecules of water combined to give 2 molecules of carbon dioxide and 3 molecules of NADH plus 1 molecule of GTP and 1 molecule of coenzyme A. What is this gibberish? Well, let me decode this reaction for you. On the left hand side of this reaction are the raw materials that are being used here, and on the right hand side are the products that are formed in the Krebs cycle. Now in every single turn of the Krebs cycle we use 1 molecule of acetyl-CoA that combines here with the oxaloacetate to form citrate. 3 molecules of NAD are used in the Krebs cycle that get reduced to 3 molecules of NADH. Here you can see where the 3 NADH molecules are produced in the Krebs cycle. 1 FAD molecule is used, which gets reduced to 1 molecule of FADH. This FADH is produced in the conversion of succinate to fumarate here. 2 molecules of carbon dioxide are produced for every 1 molecule of acetyl-CoA that enters the Krebs cycle, like I showed you earlier. 1 coenzyme A is produced in the conversion of Succinyl-CoA to succinate, where Succinyl-CoA loses the coenzyme A. And 1 molecule of GDP combines with 1 molecule of inorganic phosphate and leads to the formation of 1 molecule of GTP. 2 molecules of water are consumed, one in the synthesis of citrate and one here in the hydration of fumarate. Okay, so this was about the summary reaction of Krebs cycle. Now let's talk a little bit about how much energy is produced in the Krebs cycle. Most of the energy that is produced in the Krebs cycle is via the reducing equivalents, the NADH and FADH, that are subsequently utilized in the electron transport chain. If you look at the placement of these two biochemical pathways in the mitochondria, they lie very close to each other. The Krebs cycle operating in the matrix of the mitochondria and the electron transport chain in the membrane of the mitochondria. So the NADH and FADH that are being produced by the Krebs cycle are easily utilized in the electron transport chain for the production of ATP. Now the details of how these ATPs are produced in the electron transport chain and how many ATPs are produced are here in this video about electron transport chain on our channel. In a nutshell, you can remember that about 2.5 ATPs are produced by one NADH and 1.5 ATPs by one molecule of FADH. Like we discussed in the summary, 3 NADH are produced through one turn of Krebs cycle and 1 NADH. So 3 NADH and 1 FADH produce 9 ATPs in total in the electron transport chain. Plus we also have 1 ATP that is directly produced in the Krebs cycle. Actually GTP is produced in the Krebs cycle here, and this GTP is further utilized to convert an ADP to ATP directly. Thus, one acetyl-CoA unit generates approximately 10 molecules of ATP through one turn of Krebs cycle. Since previously in glycolysis, one glucose was split into 2 pyruvate, which were further converted into 2 acetyl-CoA molecules, which entered the Krebs cycle. So 20 ATPs are produced per molecule of glucose in the Krebs cycle. I hope you like the video. If you like our content, make sure to support us so that we can keep making videos like this. You can support us by joining our community on patreon.com or clicking the join button below. By joining our channel, you get exclusive content like handouts of this lecture and all other lectures on our channel, flashcards, behind the scenes of these videos, early notifications, exclusive posts, and much more. Thank you so much for watching.